Alright, so last chapter we learned about how things are passed from parents to their offspring. And the process that we learned was that the process is meiosis that results into gamete formation. So gametes are formed through meiosis, which is also called as reductional division. So in reductional division, the cells that produce gametes, eggs and sperm cells, and in case of plants, pollens. So pollen that we see so many during the spring season and actually throughout the summer. We keep on seeing lots of pollens. People who are allergic, they know the pollens are all the time there. So pollens are also produced through the same process, and the process is the normal cells in the gonads, male and female gonads, which have diploid number of chromosomes. They produce those gametes that have half the number of chromosomes. And the condition is called haploid and diploid. Diploid means there are two sets of chromosomes, and haploid means it is a full set of chromosomes. The complete set of chromosomes is called haploid set of chromosomes. So these haploid chromosomes that are inherited by the two parents, when haploid and haploid chromosomes, it is written as N, meaning that it is a full set of chromosomes. Male and female, they contribute these sets of chromosomes through their gametes. When the embryo is produced, initially zygote is produced. Zygote is a complete cell that has the two sets of chromosomes, just like the male or female parent where this was made from. Zygote results into an embryo, and embryo develops into a baby. So baby has diploid number of chromosomes in her or his entire body. The cycle completes like that, and this cycle that we talked about like last time, which is called meiosis, that has two subsequent cell divisions. The first cell division, which is called meiosis 1, is the reduction division. M1 is in which the number of chromosome is halved, and M2 is the one in which the number of cells is increased. So these are two different stages in meiosis. The first stage, which consists of prophase, metaphase, anaphase, and telophase, is based on reducing the number of chromosomes to half. In M1, this is what happens. In second stage, those cells that are produced at the end of M1 are doubled. Otherwise, M2 is just like mitosis. There is no difference in mitosis and M2 stage of meiosis. So the number of chromosomes that is produced, number of chromosomes that are given to the gametes is half. And another important thing that happens during meiosis is, anybody remembers what is a really, really important thing that happens? What happens actually, which does not happen in case of mitosis, is crossing over. Crossing over. Anybody remembers what is crossing over?
Two chromosomes share together.
And the two chromosomes, which are duplicated chromosomes, they cross over with each other. When they cross over, these are two duplicated chromosomes. It takes place in M1 in prophase number 1. So this shuffling or this crossover, overlapping with each other, results into those chromosomes that are different than the original chromosome. So genetic shuffling takes place here. Genetic exchange of material takes place here during prophase number 1 of meiosis number 1. So this is the source of all the variability or maybe half of the variability in this universe. This is what results into everything that we see different from each other. Even within siblings, within the same family, all the changes that we see in their phenotypes, in their appearances, is based on this one event, which is called crossing over. And that takes place in the very first stage of meiosis. So that concept you need to keep in mind. Things that we talked about last time, you need to really keep those things in mind. If you have not reviewed those after the chapter was done, then do that after you go back today. Otherwise, you're going to be lost. So the terms that you need to know from this chapter are these here. Homologous. You should know what is homologous. Then we will learn about heterozygous. We will learn about homozygous. So you also should remember what are gametes, what are gonads, what is a zygote. Then we will talk about diploid, haploid. Then you should also remember these F1, F2, true breeding, hybrid, phenotype, genotype, what else? Dominant, recessive, and of course you should know who Mendel was. So this chapter is related to patterns of inheritance. Patterns of inheritance were described first by Mendel back in 1860s. So more than 100 years ago. So Mendel's contribution to science is that he was actually a clergyman. So he actually did a lot of preaching, and he actually taught people about religion. But he was also a scientist. When he had more time, he used to do science. So he was trained in biology and in mathematics, and then he was also a religious person too. So what he explained was basically what we know today. So we see actually, if you have a cat, and that cat has a few babies, you see that all the babies are very different from each other. They look pattern-wise, they look very different. And some of them actually look like mom, and some of them don't look anything like mom at all. They look just like, if you know the dad, they probably look just like the dad. But in other cases, they are in combination of both of them. And in other cases, they don't look like either of the two parents. In case of humans, we see the same thing too. Sometimes the babies are in between, and those, sometimes people actually think that those are basis enough to feel that this baby belongs to that couple. But that's not always the case. We see that at Maury and other shows like that too, because those things are brought to the national TV, and people get humiliated there, they cry and all those things. But those are all setups actually. Those are just for their ratings. It's not really, people don't know where those babies come from. They know it very well, but it is all genetics. Sometimes babies look different than either of the two parents, and there have been cases in history where in those couples, kind of interracial type of couples, the babies look completely different. And in some cases, the white parents have black babies also, and black parents have white babies also. So, and that can happen also, though very rarely, but it can happen like that, because they had some of their ancestors that come from a different race. So this is what we will try to learn, in whatever time we have today. We need to try to finish this chapter today. So we learned about this thing last time, that the chromosomes in our body are always in pairs. In our cells, chromosomes are always in pairs except in our gonads. except what is produced in the gonads. Even the gonad cells are also diploid cells. They have two chromosomes that are inherited. In your entire body, you have two chromosomes in each of your cell, or two types of chromosomes in each of your cell, and both of those are contributed from the two parents. So two of your parents have contributed equally to make that. And when you align them up, we see a pattern like that we saw last time also, that each chromosome is identified with its partner, with its homologue, and it is called this pair is called homologous pair, or they are called homologues, meaning that they are similar, they are not identical. None of those are identical. They are very similar, but what makes them different is the person who contributed that chromosome, your father and your mother, because both of them are different, so they are different. Even though the length is the same, size is the same, sequence is somewhat similar, but they are not identical. And that is the reason, actually, all the variability that we see in families come from those. And on chromosomes, they have sequence that we talked about also last time. So the sequence on the chromosome is DNA. Chromosomes are made of DNA and proteins, two things. And sequences on chromosomes make different genes. We have how many genes? Anybody remember that number? How many genes make us? Not genes. 46 are the number of chromosomes. But how many genes are located on chromosome? Each chromosome has about 1,000 gene on it. So we have about 22,000 genes. About 22,000 genes are what define us. What make us are 22,000 genes. And those are these genes. There are some examples are shown here. And when these are duplicated, the two arms that are made of the same chromosome, they are identical. But here you can see some differences. This gene is the same here, which is the homologue of this chromosome, but the gene is different here. So this is big B, and there are no numbers, there are no letters on our chromosome. They are just molecules. So whenever you see these numbers on them, or these letters or these numbers, this is what we have given them. This is what we have kind of designated them as. Otherwise, there are no letters. They are just DNA inside the chromosome. And scientists have figured out that this part of a chromosome does something. It makes a protein, or another part makes a different type of a protein, and they can be the same when two homologues are put right next to each other, or they could be different. Like this sequence of DNA is different than this sequence of DNA. So this gene, which is this, is the same. This gene is different than this gene. So when there are two genes which are present on homologues, on homologous chromosomes, those are called, they're not called genes anymore. They are called alleles. Those are called alleles. Different forms of the same gene. It's the same gene that has different form. One is B and other may be B, or it could be big B or little b. So again, there are no letters on our chromosomes. We do not come with those labels on our chromosomes. We just designate. We just assign them some letters so that we can identify them easily. So these are two different alleles, meaning one is present on one chromosome, other is present on the other chromosome. So this concept is very important. If you don't understand this concept, then you will have a very difficult time to understand what Mendel did. Mendel really did not know about those things. What he did was, he was actually working completely blindly, because at that time there were no microscopes, or at least microscopes were not easily available. He had no idea about chromosomes. He had no idea about DNA. He had no idea about proteins. So what he did was just based on what he did with the piece. He just used the piece to understand how genetics work. Now we know all those things, but when he was working on those things, he had no idea. Now he even did not really know how the zygote is the one that has all the chromosomes. Because the concept of chromosome did not exist at that time. Concept was introduced about 40 years after what he did. So That's why actually he's regarded as the father of genetics, because what he put together more than 100, around 150 years or so now, is still applicable today. Actually, it is more obvious now than it was at that time. So homologous chromosomes are clear now. So alleles concept should be clear in your mind too. There are two chromosomes, one coming from the father side and one coming from the mother side. Both of them carry alleles, carry genes, and when we say different types of genes or different forms of the same gene, those are called alleles. And alleles could be same, alleles could be different. So one example that I gave last time also is, for example, on this location of this chromosome, these are the two homologs, these are the two homologous chromosomes. This part of the chromosome has a gene that controls eye color. Some people have brown eye color, some people have green eye color. And these are the two alleles of the same gene, because both of them control the same trait. Both of them control the same characteristic, which is eye color. But eye color could be blue, or it could be brown and green. So this person, that is this person, which is a diploid individual, carries both of these alleles on his or her chromosomes. If they are like this, when they are different, like big B and the gene should be like this, because they are different. So when this condition is there, when the two alleles are different, this condition is called heterozygous. If they are the same, if it is big B and big B, then that condition is called homozygous. Meaning two alleles are identical, and in this case, or there could be a third situation also. It could be little b and little b, meaning it's a different form of the same allele. So in that case, that is called, this one is called homozygous also, because both are the same. Both are the same. Here they are different. When they are different from each other, then that is a heterozygous condition. Hetero means different or other or variable. So whenever they are same, whether they are big letters or little letters, then they are called homozygous. So, and the genes are located on the same, because each part of the chromosomes control a different trait in our body, or in plants or animals everywhere. So each part, which is called as a gene, controls a different trait in our body. So Mendel's work that was pretty much done, all of his work was done with only one plant, which are peas. And those of you who have ever grown peas in your backyard, you know that peas are the most easy plants to work with. You know, you just put some seeds in the soil, and if they germinate, if they grow, then right in the middle of summer you will get lots of peas from them. You know, if the insects don't attack them, or if your children don't attack them, you will still get some peas from them. So the reason actually he selected this was because these are very easy plants to work with. These plants, you don't really have to do much with them. If you grow them, they will give you seeds. You know, you can harvest and eat them, or you can also control what you really want to do with them. You can emasculate them. You can actually remove their male parts of the flower, and they will become female flowers. If you take pollens from one flower and put them on the other flower, then you can actually change the genetic makeup of what will be produced from them. So this type of combination that he did, where he controlled the pollination. Pollination is, what is pollination? Pollination is basically taking pollens and using them on the female parts of the flower to fertilize that flower. So these are the male parts these are the female parts of the flower, which is the entire structure here. And around this part, which is the ovary, this part are the pollens. These are the pollen sacs that produce pollen. So pollens from here fall on this part of the female flower, which is called stigma. So from here, from pollens, they fall here, and then they germinate. They germinate in the form and they produce sperms. And those sperms go inside the eggs and fertilize those eggs. And in each side of these ovary, there are many ovules, there are many eggs, and those eggs become these peas when they mature. So after the fertilization of the female part of the flower, if it continues to develop, it develops into this pod. And this results into all of these eggs that have been individually pollinated by what was done on the female part of the flower. And he could control actually this mechanism. He would either allow them to self-fertilize them, or he will take pollens from a different flower, like this white flower, and then put them on the female part of the flower, and that way he will cross-pollinate them. He will take from a different flower and put it on a different flower, and that is called cross-pollination. When the male parts of the flower are allowed to pollinate their own female flowers or female parts of the flower, that's called self-pollination. The other part is called cross-pollination, in which different pollens or pollens from a different flower are taken and put on a different flower. So if you take pollens from white flower and put them on purple flower in peas, that is called cross-pollination. So that was one of the advantage that he actually took from these pea plants. So in case of pea plants, some pea plants are true breeding, meaning that if you take seeds from some green pods, or if you take some green seeds and you sow them, allow them to self-fertilize, allow them to self-pollinate. Self-fertilization means, pollens of the same flower fertilizing their own female parts of the flower. So that is called self-fertilization. So they will be the same. If you take green seeds and sow them and allow them to self-fertilize, when the new seeds will be made from them at the end of the season, they will be also green. If you take yellow ones and at the end of the season, they produce more seeds which are of yellow color, that is called true breeding, meaning there is no change that has been introduced in them from one generation to the next generation. So as if, in case of animals, if you take an animal and produce dolly from it, you remember that the sheep that was produced, the very first sheep that was produced through cloning process, that was a true breeding, that was a clone of the mother. There was no change in dolly compared with her mom. But if you have to take pollens from a different flower and put it on the flower, a female flower, and at the end of the season you see this type of a situation in which some of those are of one color and some of those are of a different color, that is called a hybrid, because the two different flowers were pollinated. Two different flowers were used to pollinate the flower. So the result of that is the chromosomes got mixed with each other. So that produced different types of traits. In this case, the trait is the seed color. It could be the height of the flower, or it could be the height of the plant as well. So some plants, if you take actually a tall plant and cross it with a short plant, then it can actually produce different. So if you take a tall plant and allow it to kind of pollinate by a short plant, then you will see some tall and some short plants, both. Or if you take a purple flower and cross it with white flower, you will see in the next generation that some plants will have white flowers and some will have purple flowers. So he took advantage of this type of natural characteristics of the peas, that they really don't need to be cross-pollinated, but they are good by themselves, or you can control that as well. So that is the flexibility that you need in case of these type of experiments. So when he did this type of crossing, what he saw was, based on the selection of the parents, based on the selection of the two parents that he used in his crossing, he selected two pure breeding or true breeding parents. One, for example, that would produce yellow seeds and one that would produce green seed, and then he crossed them. And then after he crossed them, he grew them one more time, and he found out that all plants that were produced in the next generation, so this is Gen 1 and this is Gen 2. In this generation, he found out that all plants that were produced from this cross were tall. They were not short, even though they, the female in this case was short. So, this cross indicates that this is male and this is female. And he observed that all the plants in the second generation from this type of a cross were all tall. There was actually not a single one that was short. So if you see crops during the springtime or during summertime, for example, corn or tobacco or soybean, if you see a field of soybean, they all look identical. They are all the same height. There is no change in them at all. And those are actually these type of plants. So the farmers, the seed that farmers get is actually is produced by producing two parents and controlling the traits that should be seen in the next generation. So when he saw that all of them were tall, then what he did was, in the next generation, in the next season, he allowed these tall plants to be self-pollinated. Tall plants which were here to be self-pollinated, and then he saw that he was able to see some tall and some short ones in the next generation. So that was third generation. That was actually 3 years. So his experiments were 3 years long. Each cross that he did were 3 years long. So what he called them, so we call it Gen 1, Gen 2, and Gen 3. What he called them was slightly different. He called this as parents or parental generation, or P generation. P generation. And the plants that were produced as a result of this type of crossing, he called it F1 generation. F1 generation is filial 1 generation. Filial means generation. Filial 1 generation means first generation that came from these two parents. And when he crossed this F1 with itself, this tall with tall, that was this plant, then all the offspring, all the progeny, or all the plant that came from there, he called it F2 generation, or F2. So P generation, then F1 generation, and F2 generation. F1 generation was all identical. There was no difference. Those were all clones of each other. So when he allowed those plants in the next season to be self-fertilized or to be self-pollinated, then he saw that in the next generation, the parental types or phenotypes or appearance. Phenotype means physical appearance. Pheno means physical. So he was able to see in the next generation that these two forms of the parents were seen in the next generation also. So none of those were like this parent, the short parent. So in this type of crossing, what he established was that the trait which is expressed in F1 is dominant trait, is a dominant allele, is a dominant trait because the short one has disappeared. The effect of the short one has been masked by an allele which is dominant. The allele which is dominant is tall because it is expressed in F1. There is no short one here. So he established from this type of an observation that short one has been masked by the effect of the tall one. That's why it is not shown here. But when they are allowed to self-pollinate themselves, then that short one reappears here, even though it was not present here. So it appeared here. So the same thing can happen in case of animals as well. In one generation, for example, in your children, they may have some traits in them that are not found, that are found in either you or your husband or your wife. And then in the next generation, they actually may express some of the traits that are not seen in your children, but are seen again in your grandchildren. So grandchildren will start seeing some of the same features that you have now, and are not seen in your daughter or in your son, but they will reappear in your grandchildren. So, and that is following the same type of pattern that Mendel found in his own experiments. So here he took two true breeding parents, P generation, crossed them, and then all of the offspring from the first generation were identical. And then he allowed them to go through another round of self-pollination, and then he collected the seeds and observed those seeds in the second generation, or he called as F2 generation, and he found out that this trait or this characteristic that was not apparent in the first generation reappeared here. So it skipped one generation here and reappeared in the next generation afterwards. So his pattern was a very specific pattern. He called this trait that appeared in F1 generation as dominant, and then the other one that did not appear here, he called it a recessive trait. Recessive trait means something that got masked, something that got overcome by a dominant one. That's why it is not shown here. So he called it a recessive trait. So now dominant and recessive are the two alleles. Those are the two traits that depend their expression on alleles. Again, he did not know that. He did not know the concept of allele because there was no concept of gene at that time. For him, these were just factors. He called them just factors. So recessive allele is one whose effect is masked by a dominant allele. And usually the convention to write these things is a dominant trait is always written with a uppercase letter. It is always written with big T or big G or big R or big A or something like that. Recessive traits are written as lowercase letters. So all of these are recessive and all of these are dominant traits, dominant alleles, which are what actually are expressed. And when both of those are present, dominant ones are expressed and recessive ones are not expressed. In order for a recessive trait, recessive characteristic to be expressed or to be shown, both of the alleles have to be recessive. So for example, if this green is a recessive trait, this one is also, this one is present in a homozygous recessive condition. This one is a homozygous condition, but it is recessive condition. Because the alleles are recessive, both alleles in this gene are recessive, so any recessive disease that is expressed has to be in homozygous condition. For example, in case of albino, you might have seen some individuals actually that don't have any color on their body. They become completely white because the color development does not take place in them. Their eyes are usually red because of lack of color development also. And that is a recessive disease, meaning the individual that becomes albino has to have both of the alleles that come from the two parents have to be recessive alleles. That's why parents are usually not albino, but they are like this. Both of them are the one that carry one allele with them, and both of these get recombined with one of the children that they have. Usually in the families where albinism is found, is usually there's one child or two children total in the families have albinism in them. Because the parents are heterozygous, because they have at least one dominant allele, so dominant allele masks the effect of the recessive one, so they are not albino themselves, but when they breed, when they reproduce, at that time, the chances are that some of their children will get the recessive alleles, and when it gets combined together, when it becomes homozygous, then it is shown in those children. So now this children, this child, when he or she reproduces, then they will give only recessive allele to their next generation. So it will continue in their families. People who have sickle cell anemia, which is actually a kind of a very prevalent disease, especially in case of African Americans and in Africa also. So that disease is also carried in generations. Sickle cell anemia, for example, individuals can be heterozygous, and if two of those marry with each other, if both of them are carriers, they don't have disease, but they are the carriers of allele, which is this recessive allele. If both of them marry to each other and both of them are heterozygous, the chances are some of their children will be homozygous recessive. And those are the children that will have this disease, and that disease is really nasty disease. Okay, so the next concept is genotype. Genotype and phenotype. Genotypes means the genetic makeup. Genetic makeup means whatever is present in our chromosome. Since we have two chromosomes, so the alleles on the two chromosomes make up the genotype. Genotype means type of the gene. Type of the alleles is a genotype. And what that genotype does to that individual is called phenotype. So that gene, since it is a gene, like here, this is a gene that has two alleles in it that makes this phenotype or physical appearance, which is yellow color. These two alleles, big T and little g, also, which is its genotype, appears in the form of this yellow color also. Why is this one yellow? Because it has one dominant allele in it. This one has both dominant alleles, so it is yellow. This one has one dominant allele and one recessive allele, so it is still dominant, it is still yellow because of this big G. Little g is suppressed, it is not expressed. So in order for this little g to be expressed, which is green color, both of the alleles have to be of the same kind, and in this case the genotype is little g, and the phenotype is the green color of the seed. Your eye colors are the same. Your eye colors are based on some genes which are either dominant or recessive. So usually darker colors are dominant, so that's why the brown color is more common in the populations. In any types of population, in any humans, brown color is more dominant. Light colors are usually recessive. So for light colors, and they're actually different things, but light colors are generally recessive over dominant colors in case of white colors. So the difference between genotype and phenotype is, genotype is the genes or alleles which are present at the chromosomal level, and what those alleles make are phenotypes. Your genes make you tall, they make you short, they make you have brown eye colors, or they make you have blonde hair, or they make you lots of different features. Make your fingers like that, make your nose like that. All of those things are the physical appearance because others can see. But I can see your eye color, but I cannot see what your chromosomes are made of. And genotype is usually hidden. It is inside your chromosomes, cannot be seen. The only way you can tell anything about those genes or your genotype is based on how tall you are or what eye color you are. Otherwise, nobody can tell what you have in your cells at the chromosomal level. So again, those three things that we talked about, homozygous dominant, these are the types of genotypes. When both alleles are dominant, it is called homozygous because both are the same. Homozygous dominant. It is written as uppercase letters. When both are different, one is dominant and the other is recessive, it is called heterozygous condition. When both of those are the same, but the allele is a recessive allele, then they are called homozygous recessive. For those features or those traits which are recessive, for them to be expressed, to have a phenotype, both of the alleles in the genotype of those have to be the same recessive alleles. So albinism, just remember that example. Okay, so Punner's career is named after the scientist who came after Mendel. So when Mendel was doing it, he was quite familiar with those things, so he really did not care much about whether people understand it or not. And that was one of the problems with Mendel. People really did not understand him when he was alive. Nobody recognized him. They thought he had just too much time at hand. So he's spending too much time in producing those P's where he should be focusing more on his work, which is to kind of preach. The scientist who came after him, his name was Punnett. So he came up with a kind of a schematic to see how things actually move from one generation to the other. So the schematic that he designed was to show things in form of this square. And that square is called Punnett square. And the square is this square. And what it is to follow what happens from one generation to the next generation. So what he did was he actually put male parent on this side, female parent on that side, and then he saw what type of gametes can be made from the female side, what type of gametes can be produced from the male side, and then cross them and see what happens in the next generation. So Manuel said, in P generation, this is what the case is, in F1 generation, this is what the case is, and in F2 generation, this is what the case is. But Punnett square said, let's make a more systematic way of seeing what's happening, because for people who don't understand genetics, this is the better way of making them understand. So he came up with this, that from this parent that has big G, little g alleles that control the color of the seed, and male also is yellow. So male is yellow because of the dominant allele here. So when he produces two types of gametes, because at the time of gamete formation, these two alleles will go to different sperm cells, because at the time of meiosis, these two chromosomes that represent these two alleles will split into two separate cells that will result into formation of these sperm cells. In case of females too, so when they produce egg cells, then egg cells can have only one of these two, because the number of chromosome is halved, and that halving event that takes place during meiosis results into these different types of egg cells, and each egg cell can have only one of the two alleles that are present in the parent. In the mother can have only one of these two. So there is an equal chance, so an egg is produced, and then sperms are produced. There are thousands and millions of sperms, and there is only one egg. So only one egg is produced. In case of plant, that's not the case. There could be multiple egg cells that are produced, and there are again millions of sperm cells or pollens that are produced in them. So, and they have equal chance. The probability is that all of these are present in the pool, and some of these are present in the pool. So one of these will get a chance to fertilize one of these two. So depends on which one actually becomes the combination. The probability is if this combines with this will result. If this sperm fertilizes this egg which is produced, then this will result. And if this one, which is a different type of a sperm cell with little g in it, if this gets a chance to fertilize this egg, then this genotype will result. If this fertilizes this, then this will result from it. And based on this genotype, you can estimate what type of phenotype will be produced from them. So this big G will produce this, big G, little g will produce this little g, big G will produce this, and this little g will produce this type of an offspring. And from here we can tell that have at least one dominant allele, big G, and in this case, both of those are little g, so this is the only homozygous recessive condition that will be the case. It does not mean that all of them will be of this kind or this kind. It could be all of them are yellow, or it could be all of them are green, or it could be some of them are green, some of them are yellow, and that can be seen from here. So this could be yellow because it has one dominant, this could be yellow because it has both of those dominant, this one will be yellow because at least one of the two alleles, or the dominant allele is there. This one is green because none of the dominant allele is there, and that is the condition for a recessive trait to be expressed, that both of the alleles have to be recessive alleles. So when he calculated the ratio of the phenotype, he found out there were 3 yellow and there was only 1 green. And when he saw the genotype, he saw there is a big T, little t, and there's big T, little t. So there are 2 of these two are the same. And then there is one little g, and there is one big G. So 1, 2 to 1. So the ratio that he came up with was that it is 1, 2, and 1. Or if you go with the appearance, if you go with the physical appearance of the seeds, then it will be 3 of those are yellow and only 1 is green. So all of this. So if you compare this with some situations in the families, as I mentioned about this disease, which is sickle cell anemia or albinism, those are all traits that run in the families. So when parents have some of those kind of alleles in them, it can be expressed. For example, albinism. So in that family where both of the parents are heterozygous, they're the carrier of this disease, it does not mean that the fourth child will be the one that will have albinism. It could be the first child that have it, maybe albino, or the chances are it may never, actually that family may never have that child that is albino, because this is all probability. The probability is the first child, if the chance of very first child, when the child is conceived, it could be the sperm that fertilizes the egg. Both of those are recessive, meaning the both of them are carrying the recessive allele. So when the zygote is made with the 2 recessive alleles. So the first child could be albino, or that child may never, that combination may never happen at all in that family. So it could be that all of them are just normal, but the risk is still there. The first child could be albino, and the same thing in case of sickle cell anemia. First child could be anemic, or it could be that combination never rise in that. So that's why it's important for those parents that carry these type of alleles. It can be easily detected now through blood testing, which one of the parents have some of these type of alleles. And it is very well known in the families. So when you start dating, to find out what your partner is, what type of genotype your partner is carrying. So it's very easy. You just plug some here or something from your partner and get them tested, so that you never have any of those conditions combined together. When we talk about a single trait, and you follow that in a cross, that is called a monohybrid cross, because monohybrid means only one trait, one characteristic is being followed. For example, here, the color of the seed is only one thing that is being followed. So that's why it's called monohybrid cross, where only one gene is being tracked, where only one characteristic, one trait, one gene is being tracked. That is called a monohybrid cross. If there are 2 things that are being followed at the same time, like color of the seed and height of the plant, then that would mean that those are dihybrid crosses, because we are following 2 things together. That's called dihybrid. So that will be color of the seed and height of the plant. So the color of the seed could be green or yellow, height could be tall and short. So when 2 traits are followed, when 2 genes are followed at the same time, that's called a dihybrid cross. And dihybrid cross is a little bit more complicated than monohybrid cross. This is a monohybrid cross. The result of that is the offspring that is produced as a result of crossing 2 parents that are heterozygous. The result will be, there will be 3:1 ratio, which is a phenotypic ratio. So what we can see, so it's 3:1 because there are 3 yellow and there's only 1 green, and the genotypic ratio will be, there is one homozygous dominant, 1 homozygous recessive, and these 2 are heterozygous conditions. So that will be 1:2:1 ratio, because there are 4 probabilities. So we're going to skip that part. So Mendel's laws are applied to pretty much everything that breathes, which is everything, plants and animals, everything. And this is a type of situation that I expressed earlier. If the 2 parents are carrier of a disease, for example, cystic fibrosis is another disease like that, sickle cell anemia, albinism, and many other diseases like that. Those are fairly kind of nasty diseases. So if the two parents are heterozygous, meaning they are just the carriers, they don't have the disease, but they have it in their chromosomes, the disease runs in their family. So it depends on what type of sperms and eggs get a chance to fertilize and produce the next zygote can result into an affected individual. Affected baby can result from the two carriers. That is why I was emphasizing that this type of situation should be known. You know, if some genetic disease runs in a family, then it's a good idea to really know what type of a genotype you are for that disease. You know, if there are cancer, for example, there's breast cancer that also has a kind of a family component also. There are some breast cancers that run in the families. So in order to avoid giving it to your next generation, it is a good idea to find out, you know, what type of a genotype you are and what type of a genotype your partner is to find out the chances or the kind of probability of children to develop those type of diseases. Dihybrid cross is shown here, much more complicated than a monohybrid cross. So here two different alleles or two different genes are being followed at the same time. The color of the seed as well as the shape of the seed. Some shape of the seeds is actually wrinkled. Now, if you have ever actually opened a kind of a pea pod, you will see some of the seeds are yellow in color, some of those are green in color, some of them are kind of shriveled. They are small. Some of them are round and big. So it is because of genetics. So if you have never done that before, so if you actually buy those seed pods and open them up and see, most of them will be green, but some of them will be yellow and shriveled also because genetics is playing its role there. There could be trihybrid crosses too. So, but all of those are kind of individual genes that are being run. So instead of doing this type of analysis, which is much more complicated, you can do 3 monohybrid crosses. For example, if there are 3 traits that are being followed, one is the, I don't know what this trait is. This trait may be the shape of the seed, this could be the color of the seed, and this could be the height of the plant. So you can do them individually and then actually come up with the product of what you're trying to see. But I'm not going to ask you questions from trihybrid crosses. So again, it comes back to what we talked about earlier in the chapter, that it is all related to what happens during meiosis. I have skipped some part from this chapter also, which is related to the sex-linked type of inheritance related to X and Y chromosome. So it comes back to the same thing that we talked about earlier. It is all based on this crossing over event that takes place in what stage? In meiosis 1, what stage in meiosis 1? Prophase 1. So crossing over takes place in prophase 1 of meiosis number 1 that results into recombining our chromosomes together. So when the two homologous chromosomes, they align in meiosis number 1, they exchange a part of the chromosomes with each other. And when those chromosomes result into formation of these gametes, eggs and sperms, then different eggs and sperms have different composition in them. So they're all different from each other. This is a kind of a combination of both. That one is a combination of both as well. So, and then it is all based on probability. It's all based on chance. You know, when the fertilization of an egg cell takes place, you know, nobody can control that process unless it is done in the test tubes. That process cannot be controlled. There are millions of those sperm cells that are attacking or going after a single egg cell, and if that happens, then that results into those type of, you know, many combinations that we see. Chromosomes that have genes on them that show all the genes, and it is also a very random process. Crossing over is also not a controlled process. It's a very random process where the entire lengths of chromosomes are kind of shuffled. They are all mixed together at the end, and when those result into gamete formations, then the combinations or the composition or the genotypes of those gametes is very different from each other. There are, you know, many exceptions to what Mendel put together. One of the exceptions is that you also see actually the colors in between. For example, anybody here is familiar with antrinum? These are actually some plants that we see during springtime. Snapdragons. What is it called? Snapdragons, right? Yes, snapdragons. Antrinum. So these are the ones actually, you will see red flowers and white flowers. When they are crossed together, they produce pink flowers. So not dominant one. In this case, there is no dominant. So these are called incomplete dominant because the red flower, red allele is shown in this next generation, but it is not red, it is pink, as if it is dominant but not completely. It is partially dominant. So the dominant allele in this case is not complete dominant. So it does not kind of suppress the effect of white flower, which is shown here as the pink color. So it is called incomplete dominant, or it's called partially dominant. Another condition is in case of our blood typing. So in case of blood typing, you are either type A or type B or type AB, or so you could be either A, you could be B, you could be AB, or you could be type O. So the people who are type A or type B or type AB, it is because of what is present on our blood cells. So for example, people who are type A, they have a kind of a protein and carbohydrate on the surface of their red blood cell that is of type A. That's why they call type A. Otherwise, there are no labels on our red blood cells that will show that, I am type A. People who are type B, their red blood cells have an antigen, which is called antigen, or carbohydrate on the surface of their cell, which is type B. The people who are AB type, they actually have both of them. So this is a combination of both of them. So earlier what we learned was that only one allele is expressed, so which is dominant allele. But in this case, both of the alleles are expressed at the same time, because these people are not A, because in this case if A was dominant, then these people should be also A. And they are not B type, because if B was dominant, then these people should be type B. But these are a different type of blood. These people are different because they have a combination of both of them together. So it means none of those are dominant, but both of them are dominant at the same time. None of them are dominant, none of them is dominant over the other one, but they are dominant at the same time, because when they're present together, they are shown, they are expressed in a different form. They are expressed as a phenotype, which is phenotype AB. These people who are mutants, type O, so like myself, so we are mutants because we don't have any of those on the surface of our blood cells. So the advantage in this case is that we can donate to anybody, because our body does not have the antigen, so nobody will attack these cells. So we can give it to these people or these people or these people, but unfortunately we cannot receive from any of them. So we cannot get blood from any of those types except our own kind. So if I'm type O positive, I can get blood only from O positive. If I'm type O negative, I can get it only from O negative and not from anybody else. These people are universal recipients. They can get blood from anybody. They cannot give it to anybody except to their own kind. But they can get it from these or these type of blood groups, but they cannot donate to anybody else. So if you are type AB type, you are just taking from everybody, but you're not giving it to anybody else. So you are just being very selfish. And of course you did not choose to become like that. These people can get blood only from type A, these can get only from type B or O. And that condition is called AB type. That's called codominance. So incomplete dominance and codominance are the two conditions which do not follow Mendel's law. Those are exceptions from Mendel's law. Alright, so find a little time, we'll talk a little bit more about this chapter next time. So even though we have one day and we have exam for that day, we will try to finish it up.
If